This method will help understand and perform lab techniques necessary to identify the activity of 2 microRNA in lung cancer cells in vitro. Lung cancer is the leading cause of cancer-related death worldwide. Here, we present techniques spanning from basic lab methodologies to more complex protocols, such as gene expression analysis and antibody protein microarray, specifically used for identifying the effect of microRNA's actions. The following methods will help us answer key questions regarding the effect of 2 microRNAs, microRNA 143 and microRNA 506, on regulating the cell cycle in lung cancer cells. We present different lab techniques like cell transfection, RNA extraction, quantitative real-time PCR, and microarray analysis. We hope our presentation will contribute on successful completing these analyses.
First, seed the culture cells in an appropriate flask sufficient for RNA and protein extraction to perform gene expression using qPCR or microarray analysis. Then incubate overnight with media supplemented with 10% FBS and 1% penicillin streptomycin. The next day, prepare the microRNA sample for transfection by diluting microRNA 143 and microRNA 506 in media at a concentration of 100 nanomolar each. Take out the flask from the incubator and remove the media. Wash with 1x